Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-level scientific article for January 2023. These are the potential questions that could be asked in Unit 5, Question 8 for January 2023. Let's begin with the article. Brain aging and neurodegeneration from a mitochondrial point of view. So paragraph 1 says, aging is defined as a progressive time-related accumulation of changes responsible for or at least involved in the increased susceptibility to disease and death. The brain seems to be particularly sensitive to the aging process since the appearance of neurodegenerative diseases, including Alzheimer's disease, is exponential with the increasing age. The aging process was defined by Herman as a progressive accumulation of changes with time that are associated with or responsible for the ever-increasing susceptibility to disease and death which accompanies advancing age. Indeed, age is the main risk factor for prevalent diseases, including neurodegenerative disorders. Paragraph 2. The brain is a high-energy consuming organ that requires about 20% of body basal oxygen to fulfill its function. Thus, it is not surprising that disturbances in brain energy metabolisms leads to disease, ranging from subtle alterations in neuronal function to cell death and neurodegeneration. Cellular energy is mainly produced via oxidative phosphorylation, they abbreviated it as OXPHOS, taking place within mitochondria. These organelles are often compared to powerhouses, providing cellular energy under the form of ATP molecules. Mitochondria produce the energy required for almost all cellular processes, from cell survival and death to the regulation of intracellular calcium homeostasis, synaptic plasticity, in neurotransmitter synthesis. However, when mitochondria fulfill their physiological functions, they can also be compared to a double-edged sword that on one hand produces the energy necessary for cell survival and on the other hand induces the formation of reactive oxygen species, abbreviated as ROS, that can be harmful for cells when produced in excess, with mitochondria as the first target of toxicity. Mitochondria and the free radical theory of aging. Paragraph 3. The free radicals theory of aging was stated for the first time by Herman in 1956 and postulates the aging as well as age-associated degenerative diseases is a consequence of free radicals attacking on cells and tissues. But how these harmful molecules are they generated? ROS derived mainly from the OXPHOS, or they call it oxidative phosphorylation, taking place within mitochondria. Indeed, the production of ATP by mitochondria requires about 85% of oxygen consumed by cells. Mitochondrial complex 4 reduces oxygen into water using electrons derived from reduced NAD and reduced FAD driven in the respiratory chain. An inevitable byproduct of electron transport chain activity is the formation of superoxide anion radicals, like this one here, mostly by complexes 1 and 3. Paragraph 4. In physiological conditions, reactive oxygen species are involved in processes such as immune response, inflammation, as well as synaptic plasticity, learning, and memory. However, when produced in excess, these molecules can induce oxidative stress, damaging proteins and DNA and inducing lipid peroxidation, with the corresponding mitochondrial structures as the first targets of toxicity. Furthermore, since mitochondrial DNA, mtDNA, is localized close to free radical production sites, it is directly in contact with those molecules and can exhibit oxidative damages. Oxidative stress can trigger cell death and has been implicated in the pathogenesis of many neurodegenerative diseases, such as Alzheimer's disease. Moving on. Redox homeostasis and mitochondrial bioenergetics in brain aging. Paragraph 5. A growing body of evidence highlights bioenergetic impairments as well as disturbances in the reduction oxidation or redox homeostasis in the brain with increasing age. Neuronal mitochondria. What makes our brain so special? Paragraph 6. The brain is a remarkable organ composed by highly differentiated cells that populate different anatomical regions. Neurons are polarized cells with different morphology according to their roles and their localization. These postmitotic and excitable cells have really high energy requirements. 1. To maintain their membrane potential, allowing the propagation of electrical signals. 2. To re-establish the ion balance after the firing of an action potential, example, via the sodium-potassium ATPase activity. 3. To trigger the release of neurotransmitters by fusion of vesicles to the plasma membrane. And 4. To allow the recapture of neurotransmitters from the synaptic cleft. Glucose oxidation is the most relevant source of energy in the brain, since other fuel sources such as fatty acid oxidation have an ATP generation rate too slow to sustain neuronal energy demand and produce too much reactive oxygen species that may cause oxidative stress. In consequence, neurons rely almost exclusively on the mitochondrial oxidative phosphorylation systems to fulfill their energy needs supplied under the form of ATP. Paragraph 7. Some studies aim to compare mitochondrial properties in different organs, including the brain. For instance, the mitochondria isolated from the rat liver, kidney, brain, and skeletal muscle showed significant and similar proton leak, but the phosphorylating systems appeared to be more active in the brain and the muscle. Differences in the rat mitochondrial proteome were also observed when comparing the kidney, the liver, the heart, the skeletal muscles, and the brain. Moreover, each organ possesses different protein composition, especially in the expression of proteins involved in their oxidative phosphorylation systems. Paragraph 8. It is important to note that neurons are post-mitotic cells with a lifespan similar to that of the whole organism. Unlike in other organs such as the skin or the liver, damaged neurons are not or rarely replaced during life, stressing the importance of protecting these systems, including antioxidant defenses to maintain the neuronal integrity and survival. Post-mitotic cells such as neurons seem to be more sensitive to the accumulation of oxidative damages compared to dividing cells and are more prone to accumulating defective mitochondria during aging. Paragraph 9. In addition, to bring a higher degree of complexity, neurons are exceedingly compartmentalized, comprising structures like cell body, axons, dendrites, and even more specific compartments that are the synapses. Consequently, a proper mitochondrial distribution is paramount to sustaining the energy requirements at specific locations within the different neuronal compartments. Thus, it is not so surprising that synaptic mitochondria, which need to sustain the energy required for synaptic activity, present functional differences when compared to non-synaptic mitochondria. Indeed, peroxide production was found higher in synaptic mitochondria of rats compared to non-synaptic ones. Interestingly, aging seems to accentuate the differences between these two populations of mitochondria. In 14-month-old rats, respiration was significantly decreased only in synaptic mitochondria when compared to 3-month-old rats. Besides, a higher susceptibility to calcium insults was observed only in the synaptic mitochondria of old animals. In non-synaptic mitochondria, oxygen consumption was not significantly affected by aging, and both populations of mitochondria generated higher levels of peroxide in 14-month-old animals compared to young animals. Continuing on. Age-related mitochondrial defects and the importance of mitochondrial dynamics in aging. Mitochondrial fusion, fission, and mitophagy. Paragraph 10. Mitochondria possess a residual genome, approximately 16 kilobase, coding for 13 proteins essential for mitochondrial respiratory chain function, which make them unique organelles carrying autonomous DNA. It appears that the quality control of mtDNA replication is not as efficient as nuclear DNA, or nDNA, resulting in an increased risk of mtDNA mutations. Fortunately, to avoid the accumulation of such mutations, mitochondria are remarkably dynamic organelles that divide and fuse in order to maintain a homogeneous mitochondrial population by content mixing, mtDNA, metabolites, and proteins. They also do quality control and distribution of mitochondria within the cell. Paragraph 11. Fusion/fission activity is also integrated with mitochondrial quality control pathways, allowing the detection and removal of aged or damaged mitochondria through a specific form of autophagy termed mitophagy. The exact mechanism underlying mitophagy, more specifically what triggers mitophagy, remains to be elucidated in more detail. Paragraph 12. In summary, when mitochondria fuse, they mix their membranes, the matrix, and intermembrane space, including all their contents, lipids, proteins, metabolites, and mtDNA. After this mixing, mitochondria can divide, sharing equally their new contents between the two daughter organelles. And when a damaged mitochondrion is detected, it is eliminated from the fusion-fission cycle by mitophagy, guaranteeing a homogeneous and healthy mitochondrial population. Thus, it is not surprising that defects in mitochondrial dynamics and mitochondrial quality control systems may lead to cellular impairment, and was proposed to be involved in the process of aging and neurodegeneration. In conclusion, paragraph 13, in this review, we aim to look at brain aging processes from a mitochondrial point of view, and we showed that mitochondria are the center of the free radicals theory by being a source and target of reactive oxygen species. The age-related increase in brain oxidative stress may lead to protein, lipid, as well as DNA oxidation, which in turn affects the mitochondrial function. When a pathological threshold is passed, this may trigger cell death by apoptosis. Mitochondrial dynamics play an important role in maintaining a healthy organelle population. Impairments in this quality control system may lead to the accumulation of defective mitochondria, as well as inefficient mitochondrial transport and distribution, again leading to synaptic and neuronal degeneration. Neurons are particularly vulnerable to oxidative insults and mitochondrial dysfunction, given that they are post-mitotic differentiated cells relying almost exclusively on the oxidative phosphorylation systems to sustain their high energy needs. Besides, distinct mitochondrial populations can be observed in different neuronal compartments, example the synaptic versus non-synaptic, highlighting the importance of a proper mitochondrial distribution in these highly compartmentalized cells. This brings us to the end of the article. We'll continue to breaking it down. To break the article down, we begin with paragraph 1. They could ask about neurodegeneration, and this could be either Parkinson's disease or Alzheimer's disease. They could also ask about investigating the human brain using scanning techniques like CT scans, MRI, functional MRI, as well as positron emission tomography scans. So please revise page 270 to page 272 of your textbook. Another part could be asked about chemical balances, again page 273 to page 275, asking you about linking Parkinson's, dopamine, as well as L-DOPA. L-DOPA can be used, of course, as a medication for people who have Parkinson's disease, because L-DOPA can cross the blood-brain barrier, and once it's in the brain, it can be used to produce dopamine, and this dopamine can help alleviate the symptoms of people who have Parkinson's. So you read about that. The other part could be asking about depression and serotonin, and again, this one here could be connected to the serotonin reuptake inhibitors, as well as drugs that could be used to alleviate the symptoms of depression. From paragraph 2, because they talked about oxidative phosphorylation, neurotransmitter, calcium homeostasis, cell survival, and cell death, I thought they could ask about oxidative phosphorylation and chemiosmosis. So I think it's a good idea for you to focus on the whole process of respiration until the end when ATP is produced. So please focus a lot on oxidative phosphorylation, chemiosmosis. You could also focus on anaerobic respiration. You never know, they could ask anything, but because they're focusing a lot on this ATP production, so more questions could be about oxidative phosphorylation, but chemiosmosis is going to be relevant as well. Then the next part, because they talked about neurotransmitter synthesis, since most neurotransmitters are going to be proteins, so I suspect they could ask questions about protein synthesis. The third part, they could talk about ATP and calcium homeostasis. So calcium is vital in muscle contraction as well as transmission of an impulse around a synapse. So I recommend you focus on that and fully understand how calcium balance or calcium homeostasis is going to be vital, mainly in muscle contraction as well as transmission of an impulse. Because remember, if we do not have enough calcium in the synaptic cleft, then the concentration that is going to fuse with the vesicles is going to be lower, and that means less neurotransmitter is going to come into the synaptic cleft, and there could be a problem with the transmission of an impulse. A question could be asked connecting how reactive oxygen species cause harm to cells. So to further give extra information, I say they cause damage to proteins. Of course, the shape of the proteins can be changed. They damage DNA, and this could lead to mutations, which could lead to cancers. They also damage blood vessels, and this could lead to atherosclerosis. Remember, this is oxidative stress caused by the reactive oxygen species. So you studied about a little bit about oxidative stress in AS biology, a little bit. And in AS biology, there are many questions that have come about reactive oxygen species and oxidative stress in connection with atherosclerosis. So you better know about that as well. Continuing on. From paragraph 3 and 4, because they talked about the mitochondria complex, oxygen and the protons used to form water, and then they talked about the reduced NAD, reduced FAD, the respiratory chain. So I think they could ask about chemiosmosis or oxidative phosphorylation in the electron transport chain. So please focus on the details of respiration again. So if you read this paragraph as well, we can see there are some advantages of reactive oxygen species they've talked about, like immune response and inflammation. Of course, the body produces these reactive oxygen species. If they are in appropriate amounts, they are advantageous. They help destroy pathogens and keep us healthy. However, if their concentration is really high, that is when they will cause damage to DNA as well as proteins and other areas within our bodies, and that's when the problem comes in. So I thought about advantages of reactive oxygen species, talking about the immune response and inflammation. Now, on that same note, I thought about specific and non-specific immune response. Yeah, it's quite trivial, but it could be asked. You never know. Then further on, I went on to say how they could ask how inflammation is triggered. So you need to focus on histamine production by mast cells as well as basophils. They could also ask about problems associated with excess production of reactive oxygen species. Here, we talk about oxidative stress, which is DNA damage to proteins, damage to nuclear as well as mitochondrial DNA. I've emphasized on this already. They could also ask how DNA and protein damage by reactive oxygen species could cause cell death and neurodegeneration. It's going to hinder processes inside the cell. Cellular activity could decrease or even stop. So protein synthesis could decrease or even fully stop. That will lead to less or no neurotransmitter production. So impulse transfer from one neuron to the next is going to be blocked or is not going to be possible. And we know if any synapses are not being used, meaning there are no neurotransmitters flowing through those synapses are going to be cleared out or they're going to be destroyed. And therefore lead to neurodegeneration. This occurs in people who have some neurodegenerative conditions or disorders. Moving on. From paragraph 5, 6, and 7, I thought they would talk about cell differentiation, how neurons become differentiated into specific cells, because they began by talking about how these are differentiated cells. If I take you back up here, they say they are highly differentiated cells, so they could talk about cell differentiation. And then they could ask how the morphology of specific neurons allow them to perform their functions, how neurons maintain their resting potential. This was focused on a lot in paragraph 6, so you need to know how an impulse is transmitted, how neurons maintain their resting potential, transmission of an impulse through a neuron, or even across a synapse, sodium, potassium, ion pumps, and ATPase activity, transmission of an impulse at the synapse. They could also ask about the events after the impulse crosses the synapse, how the neurotransmitter is recaptured, and how the impulse is propagated into the postsynaptic neuron. In paragraph 7, they said, moreover, each organ possesses different protein composition, especially in the expression of proteins involved in the oxidative phosphorylation systems. So I thought they could ask about gene expression leading to different mitochondrial proteome of the kidneys, the liver, the skeletal muscles, as well as the brain, like they stated in paragraph 7. So moving on. Paragraph 8 and 9, they talked about neurons being post-mitotic cells. So I thought they could ask questions about mitosis. Another statement I thought is they could ask why are post-mitotic cells, neurons, more prone to accumulation of defective mitochondria. In paragraph 9, they talked about structures of specific neurons. They talked about the cell body, the axon, the dendrites, and so on. So I thought they could ask about structures of a neuron as well as their specific functions. They could include myelinated and non-myelinated neurons. They could talk about synaptic activity and energy requirements. Here, you need to focus at the synapse, which processes occur at the synapse, and which of those processes require more energy. So relating ATP production and impulse transfer across the synapse, that could be asked as well. So I also thought that based on the experimental results, because you can see in paragraph 9, they talked about some experiment that was carried out. I observed that the conclusions from these experimental results were proof that aging accentuates the mitochondria differences, because we saw mitochondria of older animals generated more peroxide than those of young ones. They said 14 months as well as 3 months, non-synaptic mitochondria had the same oxygen consumption, but the 14-month synaptic mitochondria showed decreased respiration compared to 3 months. From this, we can see that aging decreases the oxygen requirements in synaptic mitochondria, leading to less respiration, while aging has no effect on non-synaptic mitochondria because 14 months and 3 months old require the same amount of oxygen. So they could ask a question about this in any form based on the data that has been presented to you. In paragraph 10, they talked about mitochondria DNA replication. So I thought they could ask a general question about DNA replication, even if it's relating to chromosomal DNA. They could ask about cell division. They could ask about mutations and their effects on proteins, enzymes, and so on. They could talk about phagocytosis in connection to mitophagy or autophagy, lysosomes containing digestive enzymes, and those digestive enzymes released either within damaged organelles, and those organelles could be destroyed. So any lysozyme activity could be asked, either in the form of phagocytosis or just as general autophagy. That could be asked, so please focus on that as well. So if we continue to paragraph 13, we can see that paragraph 13 is just a conclusion, so there is nothing else to be added on, and I don't think they're going to ask a major question about this because paragraph 13 is a conclusion of everything we've gone through. In general, this is all I think could be asked. Focus a lot on respiration, glycolysis, the link reactions, the Krebs cycle, chemiosmosis, electron transport chain, oxidative phosphorylation, the whole thing combined. You have to ensure that you know all of it. Then you think about mutations and how these mutations could affect specific cells. Then you think about, of course, the reactive oxygen species. Since they've told you how they affect the cells by damaging DNA, they could ask anything about the nervous system, impulse transfer. I recommend that you read deeply about the whole nervous system. Anything connected to the nervous system, that could be very helpful. So the general thing is deeper understanding of respiration and deeper understanding of the nervous system. So this brings us to the end of the review of this scientific paper. Thanks for being with us. Don't forget to subscribe. See you in the next video. Bye-bye.